What are those called? They're the light-dependent and light-independent reaction. And the light-independent reaction is also known as the what cycle? Calvin cycle. Good. All right. Aerobic respiration has 4 steps, and they are called glycolysis, transition step, Krebs cycle, and the electron transport system. And this table in your notes is a summary of the 4 steps, where each of the 4 steps occurs, the changes in the substrates, that is the changes in the glucose and the molecules that come from glucose as they go through those steps, and what sort of yield of high energy molecules you get, both in terms of ATPs and NADHs and FADH2s. Incidentally, these carry high energy electrons. When we talked about photosynthesis, we saw a very related carrier of high energy electrons called NADPH. It's essentially the same molecule. But it's NADH here and FADH2. They're high energy electrons. Now, this table right here, I'm intended for you to use as kind of a summary table. It encapsulates all the major information that I'm going to go through in this lecture. And so what I'm going to do is sort of skip this for now, but every time I complete a section on glycolysis or transition step of Krebs cycle or electron transport system, I'll flip back to this table just so you can see where all this data comes from. So let's start off with the glycolysis step. So notice maybe one thing to begin with, the glycolysis step does not take place in the mitochondria. There's the mitochondria. Glycolysis takes place out here in the cytoplasm of the cell. The next 3, the transition step, the Krebs cycle, and the electron transport system, yes, those do take place in mitochondria, but glycolysis is the exception to the rule. It is in the cytoplasm. All right, and the term glycolysis means sugar splitting. Glyco is sugar, and lysis means to split. And that's essentially what happens. Glucose, which is, of course, a C6 molecule, 6 carbons, in the glycolysis step ends up split into 2 3-carbon molecules, which are called pyruvates. That's the gist of it. Now, I think when I click this, it's going to show you the full-blown glycolysis metabolic pathway. It is a metabolic pathway. There are 10 enzymes involved in the glycolysis pathway, and they do this conversion of glucose into pyruvates, one small step at a time. But in the end, that's what you end up with, is 2 pyruvates for every glucose. As I click through the glycolysis pathway here the first time, it shows everything. It shows the exact structures of all the intermediates, the names of all the intermediates, and the names of all the enzymes. You don't have to memorize everything it's showing you. The only thing I want you to know is what's listed up here. But just in case you're curious to see it, here's what it looks like. Starts off with glucose. An enzyme called hexokinase adds a phosphate to glucose. And so now you have glucose 6-phosphate, and then another enzyme adds, oh no, this one doesn't add it, it switches around into fructose, so now you have fructose 6-phosphate, and now another enzyme adds another phosphate to it, then you get fructose 1,6-diphosphate. That, and then finally it's split into 2. By an enzyme called aldolase. But these are not the pyruvate yet. These are 2 C3 molecules, but they're not the pyruvates we were looking for yet. One is dihydroxyacetone phosphate, and the other is glyceraldehyde 3-phosphate, which is good old pGal that we saw from photosynthesis. Now there's an enzyme not shown on this diagram that takes the dihydroxyacetone phosphate and converts it into to pGal. And so what you end up with is actually 2 molecules of pGal at this step. There's that enzyme if it was in the diagram. So everything that's going to come after here on the diagram, we're not done with the diagram yet, but everything after this on the diagram is what happens to the 2 pGals that continue on through glycolysis. So there's the pGals. All right, so each pGal then gets taken in by this enzyme right here, glyceraldehyde 3-phosphate dehydrogenase. And let's see. There we go. You get 1,3. When they say bis, that's kind of an old term for di, and so I would say 1,3-diphosphoglycerate is the product there. Then another enzyme comes along called phosphoglycerate kinase, and you get 3-phosphoglycerate. And then one more enzyme comes along, phosphoglyceromutase. Oh, actually, I guess there's one more after that. 2-phosphoglycerate is the intermediate there. And there we go. Enolase is our last enzyme. And I think that should end up in pyruvate. No, phosphoenolpyruvate. Huh. I need to brush up on this stuff. Okay, pyruvate kinase. There we go. That's going to be our pyruvate. So, it's a pretty long pathway with the 10 different enzymes involved. All right, so what do I want you to know out of it? Well, this is a major point right here, that the first half of the pathway converts the glucose into 2 molecules of pGal. Like so. And in the second half of the pathway, the functional groups on those pGals are changed around, and so you end up with still 2 C3 molecules, but they're no longer pGals when you get to the end. They are called pyruvate molecules, like that. The other part I want you to focus in on is the high energy molecules like ATP and NADH that are involved in the process. Going back up to the start again, in the first half of glycolysis, as the glucose is getting split into the 2 pGals, at that point the cell has to use up 2 ATPs for that part of glycolysis. Now, that should strike you as a little odd, because at the whole start of this explanation of cellular aerobic respiration, I said that the point is the cells want to recharge ATPs. And here I am showing you the cells using up ATPs in the process. Well, it's an investment. Like if you heard about some company that's right about to invent something marvelous, you might invest $100 in that company, hoping that you're going to get $200 or $300 or $400 from your investment. So the cell at this point does have to invest 2 ATPs essentially to get the glycolysis process started, but there's going to be a larger payoff in the end. So in the end, it is a profit deal for the cell, so to speak. All right, in the second half, where the pGals become the pyruvates, now the cell starts getting a payoff for its investment of 2 ATPs. It gets, first of all, some high energy electron carrying molecules, these NADHs. Each one of those is carrying 2 high energy electrons. And it also gets 4 ATPs as we go from the 2 pGals to the 2 pyruvates. All right, so remembering that the cell had to invest 2 ATPs but then got paid off 4 ATPs in the second half of the pathway, the net yield is a gain of 2 ATPs for the cell and 2 of these high energy electron carriers, the NADHs, for every glucose that goes through glycolysis. Yes?
For like a cellular biologist or molecular biologist, are you going through these different metabolic pathways like individually in a graduate course or like the upper division course? And I realize that this is like an overview, or
Oh, I see what you're saying. Yeah, I guess I would say the course that's called biochemistry is where you would dissect these and the pathways in nauseating detail. I took biochemistry at San Jose State, and yeah, they do throw a lot at you in that course. You have to memorize all 20 amino acids. You have to memorize pathways where glucose is broken down and fatty acids are assembled. It's all about metabolic pathways in that course. Okay, just to summarize it again, so this is the only step of respiration that takes place in the cytoplasm. Each glucose becomes 2 pyruvates, which are C3 molecules, and the net yield per glucose is 2 NADHs and 2 ATPs. Now, oh yeah, so what happens to those pyruvates? Well, so there they are out in the cytoplasm. For the next step, which is the transition step, those pyruvates have to go to the mitochondria, and so that's where they're going to go to after this. Goodbye, pyruvates. But before we leave the glycolysis, I'll point out something else important about it. No oxygen is required for the glycolysis step to occur. And that's a bit odd because remember we're talking about aerobic respiration, which means the one that uses oxygen. Well, glycolysis is kind of the oddball step. It's the one part of aerobic respiration where you don't actually need oxygen. All 10 enzymes of glycolysis work absolutely fine without a lick of oxygen around. But it's the only step that's like that. For transition step, and Krebs cycle, and electron transport system, all of those require oxygen. Those 3 steps just will not operate if oxygen is not present. And so again, I really think of this as the oddball step. It's the only one that takes place in the cytoplasm, and it's the only one that does not require any oxygen to occur. Ready for transition step? Yes is the answer. Okay, good. All right, so look at our score sheet here for a second. Transition step is going to take place in the outer membrane of the mitochondria, where those pyruvates just drifted over to. Oh, there you see it on this diagram. Transition step is that gray arrow of the pyruvates going up to the mitochondria, outer membrane. All right, so here's my drawing of the mitochondria, and let's zoom in on the outer membrane to see what happens when those pyruvates reach it. All right, for the transition step, the pyruvates come up to the outer membrane of the mitochondria. There they are. And the mitochondria sends out to meet them some molecules called coenzyme A. Let's see, there comes some coenzyme A. Maybe a little bit about coenzymes here. Coenzymes, remember, are molecules that enzymes use to help hold molecules for them. Coenzymes are molecules that live for the most part inside an enzyme's active site, and they help the enzyme grasp substrate molecules. Well, certain coenzymes can also act as shuttles, so to speak. Certain coenzymes can grab molecules in one part of the cell and then shuttle those molecules to a different part of the cell, and that's what we're going to see here. These guys are actually going to grab some carbons from those pyruvates and then shuttle them downward into the mitochondria. So let's see what they do here. I think this one's going to start off the process. When I click, this coenzyme A is going to go up to this pyruvate. When it attaches to the pyruvate, one of the pyruvate's carbons gets oxidized into carbon dioxide, but the remaining 2 carbon atoms are going to attach to the coenzyme A, and at that point they call it acetyl-CoA. Because acetyl is an organic chemistry term meaning a 2-carbon molecule. So watch the coenzyme A, it goes over, knocks one off and oxidizes it into carbon dioxide. So the remaining two are now an acetyl group, and they attach to the coenzyme A, so we have acetyl-CoA. Now, remember, when you oxidize a carbon atom to carbon dioxide, that is a downhill exergonic energy-releasing reaction, and the cell wants to capture that energy, and so what happens is NADH capture some of that energy with some in the form of high energy electrons. And there goes that carbon dioxide. It'll eventually exit the cell and go into the bloodstream. And now the other coenzyme A does the same thing, oxidizes one of the carbons, attaches to the other two carbons, so you get an acetyl-CoA, and again, some high energy electrons are captured in an NADH molecule. All right, so that is the transition step from each pyruvate. Acetyl attaches to a coenzyme A. A couple of the carbons have been lost as CO2s. These are now acetyls. Okay, and lastly, the coenzyme A's will shuttle the acetyl groups that they've attached to